Hello, and welcome to the CAP Today webinar for Monday, April 25th. I'm Bob McGonigal, the publisher of CAP Today, and I'll be your host and moderate a question and answer session after our formal presentations today. This webinar on Monday of April 25th is entitled Innovations in Immune Profiling for Clinical Research. I'm very pleased that this has been made possible by a special educational grant from Fluidigm, and I want to acknowledge that and thank them for their support. It's very important to us. We're grateful to them. Our two featured speakers, and I'll say more about them in due course, are Caroline Roe of Vanderbilt University and Dr. Sasidhar Vemula, who's a lead scientist in Labcorp Drug Development. Dr. Vemula is based in Indianapolis. But first, before we begin, I do have the usual housekeeping items that I'd like to run over. First, we often suggest that you refresh your browser. That helps optimize the syncing of the slides and sound. You should know that live help is available throughout today's webinar. You see in the lower right-hand corner of your screen a number you can call, 858-345-5916. We have live help available to help you with any technical glitch, whether it's audio or with the website. You will see below the slides there's a box labeled Q&A, and you can type any comment in there or question that you have regarding the substance of today's webinar, but also if you have some technical questions, you can simply put them there and staff will get to them live again to help you very quickly get back to the webinar. I want you to know that you'll be able to see the full slides and audio of this webinar in about a week. We'll be posting that at captodayonline.com. And that way you can review, share with colleagues, etc. I know you're likely to see some follow-up emails, certainly from us and our sponsor also, so I wanted you to be aware of that. Finally, I did want you to know, as always, it's important that the CAP Today does not endorse any product or service that may be mentioned in today's webinar. Naturally, of course, any comments of mine are purely personal and not to be taken as policy of the CAP Today or the College of American Pathologists. So now to begin, before our two featured speakers, I've asked a special guest to join us today. Dr. Vinicius Mata is an application scientist for Fluidigm, and I've asked him to join us briefly so he can explain and introduce the very exciting technology that's at the basis of most of today's webinar. This will set the scene for our talks, and I think it will clarify for you their importance and it will help in your understanding. And so we might as well get rolling now. So I will ask Dr. Mata to begin his presentation. Dr. Mata.
Thank you, Bob, for the introduction, and thank you, everybody, for joining us today for this webinar. Today, as Bob mentioned, I will talk about our newest generation of CyTOF, the CyTOF XT, and the Maxpa direct immunoprofiling assay, which together provide an efficient and high-content immunoprofiling for clinical applications. So, okay. So I'll start off with a brief highlight of key CyTOF analytic features that make it an ideal choice for immunoprofiling. The CyTOF technology, or cytometry by time of flight, surpasses the inherent limitations of fluorescent detection by using highly pure metal antibody tags that are separate by mass instead of by wavelength. The metal tags have highly distinct signal, which can be separate with 1 resolution, as well as less variable probe intensities. In addition, there is nothing analogous to autofluorescence when using CyTOF technology, since the metals that we use as probes don't exist naturally in biological systems. What this means that very large panels can be rapidly designed and optimized that have no spectral overlap, require no single staining controls, and are extremely consistent performance from sample to sample, experiment to experiment, and site to site. These characteristics of metal-tagged antibodies provide the ideal solution to current challenges in high-parameter flow cytometry. So, mass cytometry has been adopted for use across a range of applications as the technique to deliver high-parameter single-cell analysis. Mass cytometry provides both the depth of analysis for discovery research and the reproducible data quality for translational work with a detection capability of more than 50 parameters with minimal overlap. Making high-parameter cytometry possible are accessible tools like pre-conjugate antibodies, panel kits, including the Maxpa direct immunoprofiling assay, which I'll be talking about, and barcoding reagents to multiplex samples to reduce technical variability. In the next slide, now I would like to introduce you to the CyTOF XT. It has a much sleeker design, smaller footprint, and lower installation requirements. CyTOF XT also provides a fully automated operation with a multi-sample carousel for hands-free sample acquisition. And the CyTOF XT also provides the opportunity for 24-hour operation window for increased sample throughput. Okay, and let's discuss some of the features of the CyTOF XT. So automated sample introduction, chilled sample carousel keeps sample at 6 degrees. Walk-away batch sample acquisition. The walk-away sample acquisition is also supported by a software clogging sensing, which will resolve potential clog automatically. Ready to analyze the data. The detector signal is optimized real-time during acquisition, and normalized data is ready after sample acquisition is finished. And a streamlined front-end assembly, most important, a streamlined front-end assembly with our newly designed parts make daily operations, setup, and maintenance as easy as plug and play. And of course, lower infrastructure costs. So talking about costs, here's a real-world comparison of flow versus mass cytometry demonstrated on a poster from the University of Rochester at CYTO 2019. The group compared the experiment cost for reagent acquisition processing for a 32 marker panel on mass cytometry and the same markers with fluorescent cytometry. To quickly summarize, the authors found it comparatively easier to design the panel for mass cytometry compared to flow, and also save time and run fewer samples optimizing the panel. The comparison was conducted on a previous generation of mass cytometry, so we are very excited because with the CyTOF XT, the experiment cost will be even lower in future experiments. You can also run more samples with less hands-on time with the CyTOF XT. So now let's talk about the Maxpa direct immunoprofiling system, which is a groundbreaking approach to high-dimensional immune profiling in clinical studies and overcomes many of the current challenges in conventional flow cytometry for immune monitoring. These challenges in conventional flow cytometry are multi-tube, labor-intensive assays, variability in site-to-site results, and subjective of manual gating strategy. And the Maxpa direct immune profiling system overcomes this challenge by providing consistency of an assay kit with a 30-marker panel in a single tube, plus reagents needed to process either PBMCs, or whole blood samples. This is a custom, and there is also a custom template to collect the samples using the CyTOF-XT and an automated data analysis software solution designed specifically for use of the assay, which quantifies 37 immune population in as little as 5 minutes using the Maxpa PassSet software. So combining the Maxpa direct immunoprofiling assay with the CyTOF-XT is a very powerful application for any service facility to offer and to add any research projects require longitudinal immunoprofiling. A number of individual whole blood or PBMC samples can be stained at once or individually over time. Importantly, the samples can be stored frozen because the cells are fixed and metal tags are stable. Batches of 12 samples can be run at a time with hands-free automated sample resuspension, loading, and collection with the CyTOF-XT. FCS files that are produced are immediately ready for analysis, and it can take just 5 minutes just to answer using the Maxpa PassSet software. So in conclusion, I have shown you that the Maxpa direct immunoprofiling system is a unique and best-in-class immunoprofiling solution that can meet the challenging needs of high-pressure longitudinal and multi-site immune studies, delivering answers quickly. We have proved the robustness of this assay within a site or across multiple sites participating within a study. And the comprehensive panel coupled with an easy workflow and automated analysis package enable fast, simple, and standardized high-dimension immunoprofiling, which was not possible before. So in summary, the CyTOF-XT is effective. It can accelerate clinical and translational research studies with best-in-class high-parameter cytometry. It's also fastest, providing study design and implementation much easier and quickly. And it's easy with a simplified instrument operation, walk-away sample acquisition, and important immunoprofiling solutions that include, for example, the data analysis. It's economic, minimal sample volume and reagents required, automated sample acquisition, and lower cost of ownership and installation. That I wanted to thank you for your time, and Bob, back to you.
Dr. Mata, thank you so much. That's a great introduction to this technology today for really efficient, high-content immune profiling. And I hope that you agree with me. This very well sets the scene for our next talks. And let me remind you as we go through here that at any time, if you have a question or comment, you can feel quite free to do so. Let me make sure. There we are. Oh, sorry. Here we are. Questions or comments, and we'll deal with those at the end of our formal presentations. And now it's my pleasure to introduce to you Carolyn Rowe. Carolyn is a Master of Laboratory Investigation. It's a very interesting credential. She's the Managing Director of the Vanderbilt University Cancer and Immunology Core at Vanderbilt University in Nashville, Tennessee, as you see there. And she's going to give us some discussion of a case study on cutting-edge cytometry and clinical trials. Carolyn Rowe, please go ahead and begin. Thank you.
Thanks, Bob. So as Bob said, I'm Managing Director of the Vanderbilt University Cancer and Immunology Core, which is the mass cytometry shared resource laboratory at Vanderbilt. And though we are a university core, we are nestled right in the Vanderbilt University Medical Center, and about half of our income actually comes from clinicians seeking to do mass cytometry. And we've built a pretty good framework for that at Vanderbilt. So the Cancer and Immunology Core offers fee-for-service mass cytometry. So you literally hand us a cryovial of cells, and we will provide you with FCS files all the way into analyze data and figures for your paper. And we offer 7 panels, 5 of which are human, really highlighting our focus on translational research, and then we have 2 mouse panels. And of the 5 human panels, 3 are immune-focused, but 2 are actually for other solid tumor types, lung cancer and glioblastoma, which is a brain tumor. And I highlight all this because we are available to users outside of the university community, so feel free to contact me. My email is at the end of the presentation. We'd love to help you with your research. And we also have the Mass Cytometry Center of Excellence at Vanderbilt. So we have a 3rd-generation cytoph instrument, a Helios, a little bit older than what Vinicius just highlighted, but it works great. We train users on it, and the Center of Excellence really focuses on assay development and bringing new reagents into our fee-for-service core, and I'll talk a little bit about that later. So the overarching goals of our cores that I'll talk about throughout this presentation is how we can expand clinical research using mass cytometry and how we can move what I like to term research assays, so non-diagnostic, not clinical tests, but how we can move results from those into a clinically usable space. And so today I'll talk about how we build and maintain our high-dimensional cytometry panels. I'll walk you through a case study of planning and executing a clinical correlate study, including timelines, hard numbers on samples, all the nitty-gritty details. I'll highlight the utility of patient-specific analysis tools, especially in the clinical framework, and then I'll touch briefly on how we are inferring biological significance from the results of these research assays. So this slide, you might call it the cycle of clinical research. I like to call it the wheel of pain. So you start at the top, of course, with your patient who's undergoing treatment of some kind at Vanderbilt. And so as we move through this circle, I've highlighted around the outside a couple questions that we might seek to answer at different phases in this cycle. So we have our patient, we collect blood or tissue or what have you over time. We do high-dimensional phenotyping with mass cytometry. And even before the samples get to the machine, we can already learn a lot about our collection pipeline and the usability of samples. They were shipping samples from an outside institution. This can be very informative before we even acquire any data. And then once you have your high-dimensional immunophenotyping data, we do single-cell analysis. So we'll do dimensionality reduction. We'll look for changes over time, finding cell types that change over time, and characterize phenotyping these cells. And here we can already determine the feasibility of our mass cytometry panel. We can provide proof of concept data or preliminary data for a grant to procure further funding for a larger cohort. And as we move our data through the analysis pipeline, we tailor that pipeline to answer the questions that we have for it. And then finally, hopefully, we want to correlate the outcomes of our research with some clinical marker. These are the big money questions, right? Is there a biomarker of response that we can detect? Or better yet, can we detect patients that will or will not respond to therapy and maybe save people some side effects of treatment that they won't even respond to anyway? Can we identify really aggressive disease types versus diseases that maybe we can watch and see and treat more conservatively? I think a lot about personally of immune stability. How healthy is a patient's immune system going into treatment? Are they able to respond in a way that will be beneficial for treating their disease? And then finally, for the chemist, what's the biology of treatment? How is your molecule acting on the immune system? And so obviously with mass cytometry, we answer this through panel design. And with many markers, you can do many things. You can immunophenotype your sample. Of course, that's what people usually think about when they think about mass cytometry. You can deep phenotype your cells. Now in the age of immunotherapy, cell-cell interactions have become very important, right? There's no panel is complete without PD-1 and PD-L1. But there's also other cell-cell interactions to think about. You can think about what's going on inside the cell. How are cells responding to maybe these cell-cell interactions or the drug through phosphorylation events and proliferation within the cell? And then finally, beyond the immune system, what are the other cells in the sample? Maybe you're looking at a solid tissue that has tumor cells and stromal cells. What are these cells doing in the context of your disease? And so we think, what makes a good panel, right? You want to phenotype all the known cells of interest, of course, but also with 30, 40 markers, you can certainly provide some information on unknown or unexpected cell types. And especially in cancer, all bets are off in terms of what cells are going to express. You like some non-phenotypic readouts. Are the cells proliferating? Are they dying? Are they activated by treatment or by other cells in the sample? A good panel is suitable across multiple sample types, at least immune cell types, maybe bone marrow and blood, but I mean, you can use these panels on anything. We do a lot of solid tissue work and get really great deep immunophenotyping data from those assays. And then from a technical standpoint, it'd be nice if your key markers, so maybe your blood cell lineage markers, align across panels so that you can kind of compare results from panel to panel. And then, of course, in the core, I think a lot about how can we keep a panel current or make an existing panel suitable for a new project. And it's really quite boring. Keep it simple. Build on an existing panel that you know works. I know Vinicius talked about that kitted lyophilized panel that they use to great success. Maybe there's spots you can slot in antibodies in there. You can add new reagents, of course, to get new channels to build your plexing. And I'll talk about that a little bit, about how we added cadmiums to this existing panel. And then finally, with precious clinical samples, we're always trying to find ways that we can do fewer panels, learn more with less, I guess, because you might have a limited number of samples or cells, and so fewer panels might give you a better outcome. And so for the study I'll be talking about today, we tailored a 45-dimensional CyTOF panel that I'm quite proud of. And it was designed, of course, for immune monitoring, so it really focuses on T-cell subsetting. But there's also quite a bit in here. There's a bit of macrophage information. But so here in the middle of the slide, we have a Vizzini map. So it's a two-dimensional representation of this 45-dimensional data. And each dot is a cell, and the cells are color-coded according to what population expert believes they belong to. And those colors correlate with little cell cartoons around the outside of the diagram. And then every marker from the panel is listed around the outside, and it's kind of categorized into a section that made sense to me. But of course, we can look at all these markers on all the cells. So for example, under B cells, I have CD20, which of course is a classical B cell marker, but is also expressed on recently activated T cells, and we can certainly look at it in this context. So we have a host of major lineage markers there at the top, of course, CD45 for all our leukocytes. We included CD66B. This plot and all the data I show today will be peripheral blood mononuclear cells, so there will be no granulocytes, but we included it in there so that we could look at whole blood if ever we had a study like that. CD19 and CD3 for your B and T cells, CD4 and 8 for your T helper and cytotoxic T cells, and then CD14 and 16 for classical and non-classical monocytes. And in this panel, we're subsetting our T cells based on surface chemokine receptors, so CCR4, CXCR3, and CCR6. We also see our Th2 in there for our Th2 cells, and TCR gamma delta for those specific T cells. And there's no FOXP3 in this panel, but we were able to identify Treg-like cells, so there's CD25 high, CD127 low. Activation and memory, of course, RARO, ICOS 41BB, OX40. Like I said before, no panel is complete without checkpoint, so PD1, PD-L1, CTLA4, and TIGIT. And then there's a host of what I term life and death markers that will be very interesting in all the cells. So we're measuring total DNA, Ki67 for proliferation, membrane permeability at the beginning of the assay, so what cells were dead before we even began staining, cleaved caspase 3 for apoptosis, and FAST. And then a whole host of different receptors for cell navigation. We can detect hematopoietic stem cells with CD117 and CXCR4, NK cells, and macrophages, of course. And this panel was really only possible with the addition of 6 monoisotopically tagged cadmium antibodies. So here at the top, I have that same healthy PBMC from the prior slide. And we tagged that module of lineage markers, the 66B, 3, 19, 14, 16, all of those with these cadmium tags, which I think this was part of our reagent development with Fluidigm. So these might have actually been kits from R&D at Fluidigm before they were commercially available, but you can buy these now. And you can see we get quite good signal. So on the left, we have all the live cells from a healthy PBMC, and I'm showing CD3 on the x-axis, CD19 on the y for T cells versus B cells, and those T cells subset quite nicely into CD4 positive and CD8 positive, with really minimal +1 bleed and minimal isotopic impurity between the cadmium. So that's really nice to see. And then of the cells that are not B cells and not T cells, I've pulled those all the way over to the right, and we've got CD14 versus CD16 for classical and non-classical, and then intermediate monocytes. And the staining is a little bit lower, about 1 log lower than we usually see with lanthanide tag antibodies, but it's more than sufficient for downstream assays. And that said, cadmium channels are most suited for bright targets. They're at the very low end of the mass range, so they're a little bit dimmer, but they're great for these, CD3 is so bright. And then it should be noted that 106 cadmium does overlap with the palladium barcoding kit that Fluidigm sells, which is why we put CD66B on that channel. We don't often do whole blood, so this was something we could drop if, say, we were barcoding PBMCs. And so once we had this panel, we needed something to test it on, right? And so this is actually a long time in progress. So we worked with 2 VUMC clinicians, Katie Beckerman, who's an assistant professor in the Department of Medicine, and Dr. Lior Horn, who has since left Vanderbilt for AstraZeneca. But we've been working on this project, for almost 5 years. We first discussed this clinical trial, this phase 1/2 with nivolumab and vorolanib in 2017. And we did a 6-sample PBMC pilot in mid-2018 with patients who were not on trial, but were receiving standard of care anti-PD-1 therapy, just to test the sample collection and processing pipeline to make sure that once we did get samples from the study, they were high quality and would go through the assay fine. So this study our cohort ended up being 32 patients who were receiving this combination therapy, so anti-PD-1 therapy, and then vorolanib is a anti-VEGFR, PDGFR, actually it's a tyrosine kinase inhibitor, so an older class of drugs, comparatively. And we ended up with 32 patients who had thoracic tumors resistant to standard treatment who were on trial. And these patients had a mix of tumor types. There was mostly non-small cell lung cancer, but we had a few small cell lung cancers, and hence thoracic, we had a few, we had 3 thyroid cancers. And these were thyroid malignancies, not, a goiter or some kind of non-malignant growth. And patients gave blood immediately before the start of treatment and then around day 28 of treatment. And it took about over 2 years to collect all these samples. And they were viably cryopreserved by our clinical trial processing core, according to a protocol established by our group, actually. And we had about 4 million cells per tube, and the median viability was 90%. So these are both great. We usually hope for 5 million cells per tube at 90% viability in order for successful staining and data collection. A number of samples did have suboptimal cell counts and or viability, but were included because there were only so many patients and samples were precious. And we characterized suboptimal as less than 1 million cells per tube and or less than 90% viable at freeze. And these samples tend to not perform as well downstream, but it's up to the investigator if they'd like to continue with them. And so I have this slide just to kind of hit home how many tubes we're talking about and how much work goes into even a small clinical correlate study. So we have 32 patients with 2 time points, which is 64 experimental samples here, the orange tube. We elected to not barcode these using the palladium barcoding kit, in part because of sample viability concerns that I highlighted on the prior slide. Dying cells don't barcode well, and you run the risk of ruining data for a large number of samples if you have, one rotten apple in the basket. This barcoding would also overlap with the 106 cadmium, which this was PBMC, so that wasn't a huge concern, but we did want to at least test out the antibody. And our collection timeline, we were actually still collecting samples when we had run our first, I think, 2 batches. So we didn't actually have all the samples in hand at one time, which is not uncommon for a study like this that's ongoing. So we elected to run 5 batches of 6 or 7 patient pairs, so making sure to run the 2 time points from each patient together. And then the customer provided 1 blood apheresis control per batch to kind of look for staining or any kind of batch effect over the course of acquisition. And then we also included what we call it the cover your butt control, the healthy PBMC with that first batch, just in case the sample collection pipeline is horrible and the customer provided control is also not high quality. At least we have something that will stain and we can say, you need to look at your sample collection. And so each batch was thawed, stained, and acquired together. And one thing that I would change if I could go back in time was we prepared the antibody cocktail fresh for each batch. So this is a liquid cocktail, so we had to pipette it by hand each time. And I would have liked to gone back and at the beginning of the study prepared enough cocktail for all 5 batches and frozen it down. There's a protocol I should have referenced, but anyway, we prepared it fresh with each batch, which did lead to a little bit of batch effect in staining across batches. And samples were stained and acquired over the course of about a year. And when it was all said and done, we had run 70 tubes. And of these 32 patients, 25 had enough data for first pass analysis, which we consider 5,000 live single-cell events per time point. So in each time point, at least 5,000. It's not saying that the patients who didn't have enough can't eventually be analyzed. We could find a population of interest and go back to this data and manually get it out, but for this first exploratory pass, we wanted at least 5,000 live single-cell events per time point. And as perhaps you've experienced, some patients had pretty dramatic shifts in their immune system during treatment. And others had very, very stable immune systems over this time period. So here on the left, we have patient 123 and then patient 159. And the y-axis in all these plots is CD45 versus CD3 and then CD11B. So we're looking at T cells and then monocytes/macrophages in these PBMC samples. And patient 123, I think you'll appreciate that the plots pre-treatment and on treatment, so the rows, look essentially the same. There's some shifts in staining and maybe abundance. Probably if you gated it out, it'd be a little different. But overall, they look pretty consistent over the 28 days of treatment. In contrast to patient 159, where they're almost completely losing their T cells, and then as well as their myeloid cells, or at least they're shifting in phenotype pretty considerably. And so with 25 patients, we're kind of wondering, are there any common themes in these changes across individuals? And then, of course, beyond that is, can we detect who is responding to treatment and who isn't? So to tease out the similarities in these changes over the course of therapy, we used a tool designed in our group by Sarah Vemula called TREX, and this stands for Tracking Responders Expanding. So it takes samples taken over time, so say before treatment or on treatment, as in this case, and then looks at the high-dimensional data to see where are cells emerging over the course of therapy, what phenotypes are becoming more common, and then what phenotypes are going away with therapy. And so this addresses kind of an interesting biology gap where we don't know what cells are going to be changing. We're not sure, anti-PD-1, often you look for your CD8 positive, PD-1 positive, Ki67 positive cells that are responding to treatment. But that's not the case with novel drugs. You don't know what cells are responding to treatment. So it helps to have something that is a little unsupervised in that. And this tool is also patient-specific, which is a really neat way of getting around that batch effect concern. So rather than looking at all the pre's and all the posts from all the patients, we're just comparing one patient to themselves over time. So any staining variability or machine variability from day to day is taken out because those samples were stained together and acquired on the same day with the instrument. And so all this work is driven on the hypothesis that areas that change regularly, so across patients, will include those treatment-specific cells. And this tool is especially good for very, very rare cell subsets. So we're looking at even less than 1% of the sample. And so this is the output of T-Rex. So on the left, we have two UMAP plots. UMAP is also a two-dimensional representation of the high-dimensional data. It just emphasizes large differences, so it kind of pushes your islands, if you will, of cell types further from each other. And the top is pre-treatment, and then the bottom is on-treatment. And what I think you'll appreciate is there's some differences in the pre and the on-treatment maps, as we'll call it. That dotted line is actually at the exact same spot on the two plots. And you can see that island on the right is shifting from the top, which is kind of, I'm pointing at that blue arrow, and it's shifting down below that line after treatment. And on the right, this blue and red plot, each dot is a cell. Blue dots are areas where the map is less dense, less abundant on treatment than before treatment. Red areas are where there are more cells after treatment than before treatment, and then gray is equal across time. And so those areas that I pointed out with the arrows, that large island is the island of monocytes or macrophages. And I hope you can appreciate that. So there's the label on the top is just the phenotype of the cells. So I hope you can appreciate that the top island, the cells are CCR7 positive, and then the bottom, they're shifting over the course of treatment to the CD62 ligand positive monocyte phenotype. And T-Rex also puts out an overall degree of change for the patient. So this is patient 101, and their overall degree of change was 7.5%. So over the course of therapy, their blood changed in phenotype 7.5%. And in a cohort of this size, you, of course, have a range, or perhaps not a course, but you do have a range of changes in the immune system over the course of therapy. So that patient we were just looking at, patient 101, is the last patient in the second row, and they're right in the middle of the pack with their 7.5% change. And then a few slides ago, I showed you a patient that changed not at all. That's patient 123, and they are in the bottom row in the blue square, and they only changed about 1.5%. So that's just barely more than you would expect a healthy person to change over the course of a month. And then that big changer, patient 159, is in the upper left. They're our biggest changer, and they have 75% change over the course of therapy. And all of these degrees of change are plotted in the lower right. So you can see most people are changing less than 10 or 20%, and then we have a few outliers that are changing quite a lot. And the question is, what's driving that change, and are these patients similar, and are they responding or not responding to therapy? And so just briefly, so these big heat maps can be a little bit overwhelming. But essentially, so we're doing a per-patient analysis. So from each patient, we're identifying populations of cells that are changing over therapy. They're either contracting, so they're going away with therapy, or expanding. They're becoming more numerous over the course of therapy. So in these heat maps, each row is one population from one patient. And they're compared to each other. So where a population is compared to itself, you get a red square down the midline. So it's reflected across the midline. And here we're seeing how are these populations that are either contracting or expanding in the patient similar to other populations from other patients, and are there any common themes in the phenotypes of these cells? So I hope you can appreciate in the contracting populations, there's that big block of orange, red in the middle. So that's perhaps a large number of populations from different patients that have a similar phenotype. And so using this analysis, we were able to identify 56 clusters that are changing over the course of therapy, clusters of populations that are similar across patients. 23 of these were contracting, 33 were expanding, and we're in the process of kind of combing through these clusters of populations, seeing if they're expected, if they're novel, and if there's any correlation with response to therapy. but that work is ongoing. So in summary, we walked through panel design for human immune monitoring. I introduced you to my clinical trial. We looked at using analysis tools to tease out per patient changes during the course of therapy. And then I briefly touched on aligning results of this analysis across the patient cohort. And with that, I'd like to, of course, thank my lab, the collaborators, the patients for this generous donation of their samples, and our funding sources. And I'll hand it back to Bob, I guess.
Bob, your microphone may be muted.
Carolyn, thank you very much. That's an incredibly good presentation of using this in real time on a case study for a clinical trial in important cancers and important drugs. And I think we're all blown away by how detailed the information can be. So as we go through here, let's go ahead and bring up Dr. Vemula's slide. Very good, thank you. Remember, we'll have time at the end for some questions and answers. If we don't get to all of them, our friends at Fluidigm will certainly help you get your question answered. But now it's my pleasure to introduce our third and final speaker today. Sasidhar Vemula got his PhD and is now Director of Translational Biomarker Solutions. He works for Labcorp Drug Development, and he's going to speak about some method validation studies using the Maxpa direct immune profiling assay. Dr. Vemula.
Thank you, Bob, for your kind introduction. Sorry. Good afternoon. I would like to take this opportunity to thank the organizers of CAP today and Team Fluidigm for giving the opportunity to showcase the Labcorp Drug Development and Translational Biomarker Solution capabilities and state of technology in the clinical research arena. After listening to two excellent talks on case studies, clinical case studies, and instrument technologies, today's talk is focused on mass cytometry-based method validation studies using the Maxpa direct immune profiling in the PBMCs. So here are the outline of my presentation today. First few slides on the mass cytometry and the Maxpa direct immune profiling assay overview. And Maxpa, PathSetter, and Cytobank, these are the analytic software we use for analysis. And the qualification parameters for mass cytometry assays, and the method validation experimental plan, and mean frequencies of the TB, NK, and myeloid cells, and finally assay qualification criteria. Mass cytometry is one of the variations of flow cytometry in which antibodies are labeled with the heavy metal ion tags rather than the fluorochromes. Mass cytometry powered by the CyTOF technology, also known as the cytometry by time of flight. CyTOF allows the phenotyping of immune cells based on 50 different cell surface and intracellular markers simultaneously on the million of individual cells with a minimum spillover between the channels. The outline of the mass cytometry approach is here. This includes the panel design, staining, acquisition, and analysis of high-dimensional data. New flow cytometers with 30 different channels make a compensation matrix increasingly difficult as the number of channels grows. Due to the unavoidable overlap between the emission spectra of the fluorescent dye, as seen in the left figure, there was a minimum spectral overlap in the mass cytometry as is seen in the right figure. The Maxpa direct immune profiling system consists of Maxpa direct immune profiling assay and Maxpa PathSetter software. The Maxpa direct immune profiling is a highly multiplexed 30 antibody marker panel which allows the comprehensive immunophenotyping of human PBMCs or whole blood. As seen in the cartoon on the left, 37 immune cell subsets such as the TB, NK, and the myeloid cells were identified in the PBMCs and the whole blood. Other key benefits include efficiency with a simple and single tube workflow, reliable as it can produce a consistent result in the lot to lot, run to run, and site to site, and flexible by design. We can add up to 18 antibodies to support our study goals. As custom antibodies which are added to open to the channels in the MDBA panel as seen in the right panel. The minimum channel crosstalk in the mass cytometry simplifies the expansion of the high parameter panels, which is the significant advantage over the fluorescent cytometry. With the open channels, it supports the custom conjugation. Additionally, we can also label the own antibodies using the PhDm Maxpa labeling kit. The Maxpa assay is a fully automated reporting and data analysis solution that automatically identifies 37 immune cell types in the FCS files from the sample processed with the Maxpa direct immune profiling assay. In the software, eliminates the variability of manual gating and also provides an efficient and reliable solution for longitudinal and multi-site studies. Maxpa Assay provides an automatic selection of and enumeration of single viable cells and staining quality assessment for all the 30 markers and 2T dot plots and the high-dimensional Disney plots for identified population, and a report summary which gives the cell number or percent of the total cells and the percent of the parent population, and lastly QC report matrix for the acquisition and the model quality. Besides the Maxpa processor, the Cytobank Enterprise license is comprehended with API applications. These slides demonstrate the high-dimensional mass cytometry workflow at the Translational Biomarker Division of the Labcorp Drug Development. The Cytobank is a cloud-based data analysis software that turns the high-content data into the high-impact knowledge by providing the tools optimizing for the mass cytometry, including clustering the SPADE and Whitney, heat maps, dot blocks, and histograms and statistics, along with the ability to share the data with the sponsors and the collaborators. The TBS uses the MGPA to validate the clinical samples under drug development as is seen in the images below. The table indicates isotope, metal tag, and antibodies and clone information included in the MDPA in the dry single tube format. Another panel we have extensively used to validate the clinical sample is the Maxpa basic human T-cell immuno-oncology panel. Panel details are mentioned in the table. And here is the method qualification parameters for the mass cytometry assays we adopted. The intra-assay precision and inter-assay precision sample stability are the three important steps we consider for method validation. The first, intra-assay precision, in which 4 donor samples are run in triplicates, and the acceptance criteria is below 25% of the coefficient variations. The inter-assay precision, same donor samples which were run in triplicates by 2 technologists, with the acceptance criteria also is below 25% of the coefficient variations. The sample stability has the 4 normal PBMC samples assayed in the triplicates and freshly prepared as the baseline, which being stored at liquid nitrogen tank for 30 to 60 days, and established at least the time point where the minimum 20% of changes from the baseline or changes in the lesser than 20% of CV achieved the minimum 80% of the samples. To developed the mass cytometry assay. So we check PBMCs are stained with the assay tubes according to manufacturer instructions on day 0. On day 0, samples were stored at 4 degrees and acquired them on the site-off, while remaining the samples were stored at -80 degrees at the indicated time period. The assay tubes were subjected to the data acquisition on the Helios site-off tool, and FCS files were exported and analyzed using the Maxpa setup software. Here, we show the mean frequencies of the T cell subsets in the PBMCs. 4 healthy donor mean frequencies of T cells and the subset population were shown in the line graph. X-axis represents the mean frequencies of T cells and its subsets, and Y-axis represents the kinetics of the PBMC stored at -80 after staining on the day 0. T cell subset expression was the maintained at the indicated time points. And again, on the slide, this shows the mean frequencies of CD4 helper T cells and the CD4 naive cells and CD4 central memory cells at day 0 to day 60 time points. And the mean frequencies of the TH1, TH2, and TH17 cells, and gamma and delta T cells and NKT cells are remain unchanged when compared to the 0 on the day 60 time point of cryopreservation. That means these receptor levels are unchanged. And we also show the mean frequencies of the B cell subsets at the day 0 to day 7, day 14, day 26, and day 60. And the B cell expression pattern also is remain unchanged upon the cryopreservation, as is shown them in the line graphs here. B cell naive population, B cell memory population, and plasmablast population too. And the mean frequencies of NK cells from the 4 healthy donor subjects are also shown here, and they also remain unchanged when compared to the day 0 to the day 60. And here we show the NK cells early and the NK cells late cells based on the expression marker of CD56 expression. Mean frequency of the monocyte subset and 4 healthy donor PBMCs are shown here based on expression of the CD14 and CD16. And monocytes are shown there, and monocyte classical monocytes, which are the CD14 positive cells, and the transitional, which are the CD14 and CD16 intermediate populations. So we did not see much difference when compared to the day 0 and day in all the 4 donor populations. And another immunocells, which is the antigen-presenting dendritic cells, subsets also shown them here, with the plasmatoid dendritic cells and the total dendritic cells and the myeloid-derived dendritic cell based on the expression of the CD123 and the CD11B. And here also, we did not see much difference in the expression pattern of the storage in the -80 degrees for a indicated time points. And we also look into these very rare populations like basophils and eosinophils and the granulocytes and the neutrophils. And we do see the difference in the patients and the variations, but in the overall the variation is not in a significant levels. So to qualify this assay criteria, so 2 technologies run this assay. We use the same PBMCs, and here on the left side we indicated here on the left we indicated the immune cells, and here the percent of the CVs for the run 1 and run 2, and these are the grand mean CVs. So if grand mean CV is greater than 25, that means this assay is not qualified. So all the grand mean CVs for this immune cells shows that below 25% of the CV. And whereas these populations where, we do see a very low frequency number of the cells, basophils and eosinophils in the eosinophils, which shows about 25% of the CV. That means, for the rare populations, the acceptance criteria for the approved assay is the 35% of the Granville series. So in summary, we validated a Maxpa direct immune profiling assay kit using the PBMCs. Assay is qualified for the clinical sample testing. So here I present, TBS, the Translational Biomarker Solution mass spectrometry services. We validated Maxpa direct immune profiling assay, MDPA kit, and validated Maxpa, where we are validating Maxpa basic human T-cell immuno-oncology panel. And we also designing the experience in the designing the customized panel. And we also have the capability for the, target tissue isolations. And we can also add the intracellular cytokine markers or, transcriptional markers to the same panel. And we also have the capability of doing the barcoding with the help of the Fluidem people. And I thank you for your attention.
Dr. Vemula, thank you very much for that presentation. You've shown us yet again another wonderful example of how to validate this immune profiling with this technology. We thank you very, very much. Now, of course, we don't have a lot of time left, but I'd like to do a couple of questions and answers if we can quickly. I also want you to know that your question and even your name is something that we've captured, and in many cases we'll be asking our friends at Fluidem to help follow up with you to answer your questions. Let me, though, begin first with Dr. Mata. I have two questions. We heard a lot about samples. To get a complete profile for a patient, what is the recommended sample? That would be blood, I believe, and then what quantity of blood? Dr. Mata.
Thank you, Bob. That's actually a very good question. The Maxpa Direct Immune, I'm going to speak specific about the Maxpa Direct Immune Profiling Kit. So how you design it for easy to use and which kind of cells and how many cells you use. But be aware, like as Carolyn mentioned, you can design your own panel. You can have flexibility to whatever cells you want to use in the cytoph technology, right? So we are not strict to what I'm going to say here. I'm going to say here specific for the Maxpa Direct Immune Profiling Kit, which makes a clinical trial very fast and easy to be carried out, as Dr. Vemula also mentioned about his validations. So we have designed this kit to be used with 3 million PBMCs or just as few as about 300 microliters of whole blood. So you can use fresh whole blood, add directly to the tube, incubate for 30 minutes and perform the protocol for lysing red blood cells and fix the cells before excision. Or you can prepare your PBMCs and then add about 3 million cells into the tube with about a total volume of 270 microliters. So that's, if you need more information, we are happy to share the protocol and more details about how to prepare your samples. But that's as easy as it can get, just 3 million PBMCs or about 300 microliters of blood.
Thank you very much. Tell me briefly, how stable are the metal-tagged antibodies in this technology? What is their shelf life? Do you store them in the refrigerator or do you store them frozen?
Yeah, we usually keep the antibodies just at 4 degrees. And the length the antibodies I use, probably Caroline can also chime in. They have probably experience. So we as a company, we provide 6 months to a year, like storage time, expiration date. But if you look at our other users have been using it, and the Cytoforum is like a community list where people discuss. So there are users, customers using antibodies that has been labeled with the metal tags for more than 2 years or 3 years. And I think it comes down not to necessarily the stability of the metal tag conjugation, but the quality of your primary antibody. So if you conjugate the antibody yourself, so the primary antibody is good shape, you probably can have it longer. But that's what we hear. But we as a company, we have to set a date of 6 months to 1 year, depends on the output.
Very good, thank you. And let me ask you, Carolyn Rowe, talk about the ability to actually do a functional analysis of the various immune cells using this technology, CyTOF.
You mean like cytokine expression, or what kind of function? Because you can certainly culture them, and then we do a lot of what we call phosphoflow in the lab, so we'll culture cells with some kind of stimulating factor, and then you can fix the signaling in place and then measure phosphorylation events on proteins like STATs and ERK and that sort of stuff with antibodies. So if there's an antibody for it, you can certainly measure it.
Okay. Is that typical in your lab to have those questions arise?
Yeah, I mean, for a clinical study like this, I would generally prefer to do just phenotyping and then maybe a Ki-67 or a cleavecast-base just for that life or death, rather than subjecting the cells to some kind of ex vivo stimulation to look at their function. But you certainly can do that. I've done it. I actually published a starter protocol on that in 2020. So.
Very good. Dr. Vemula, the same question to you. How important is the ability to make some functional judgments about the various immune cells identified?
Yeah, thank you, Bob. And we mostly, we do the immunophenotyping, and at the same time, some of the sponsors, they want to do the know the transcriptional factors and the phosphoflow, and at the same time the cytokine too. And we are also have the capabilities to do those assays in the immune cells.
Very good, thank you. Carolyn, tell me, what is the latest? Are you envisioning doing a great number of case studies and clinical trials similar to the case you presented so well today for us? Great demand?
Yeah, it's really up to the investigators at Vanderbilt and whoever comes to us with a problem. I found I've not had to go out seeking that sort of stuff. It just comes to me. So I try to focus on keeping our services as up to date as possible. So we're debuting the cadmiums for all Cancer and Immunology Core panels now, so that immune module will slot into everything. And we're working on building out our solid tissue panels. And then I've got my eye on adding 4 more channels with monoisotopic cisplatins in the next couple of years. So that's kind of the technical areas where I focus on.
Okay. And give us just a little more color on discussion of the T-Rex populations. That's a very fascinating set on your slides. We didn't have time. We could probably make an entire program just on that. But talk a little bit.
Yeah.
I bet you have. Talk about some of the common themes in immune response that you see from the patient's data when you put that lens on the data.
Yeah, so we've so that algorithm is newer. It's probably 3 years old at this point. It got published last year, I think. So we've applied it to a lot of different contexts and found really interesting things. The initial application was actually spectral flow data, not CyTOF data, out of UVA, and looking for antigen-specific T-cell responses in viral infection. So it's been really cool for, rather than staining with tetramers, you can kind of predict the phenotype of your T cells that are responding to therapy and find them without the tetramers. What else? We've just been applying it to different solid tissues in different contexts. I don't know that there's a common theme yet, because we're kind of working on getting a breadth of examples with that algorithm.
I see, sure.
Yeah.
Now, is this something that you also share? In other words, do you have a working group of people using the Maxpa software for analysis that you share data or at least experience with?
We don't use the Maxpa path analysis, but all of our, I've tested it for Fluidigm. It's perfectly fine. We just like having a little more bespoke analysis. But all like T-Rex, all of our algorithms are available on GitHub, and most of them are on bioRxiv as well. And if you have a hard time finding any tool that you might like to use, certainly reach out and we'd be happy to share it.
Very good. Thank you so much for that. With that, I guess I'll give you the last word because it is time to wrap up. You've all been very patient, and you've been a wonderful audience staying with us throughout. So to sum up, I would like to thank Dr. Mata. I'd like to thank Carolyn Rowe and Dr. Vemula for 3 wonderful talks today. I would like to thank Fluidigm. This was made possible by their special educational grant, and we're very grateful for that. We want to acknowledge that. We encourage that sort of partnership and sharing of information. So I want to thank them. And finally, I want to thank all of you because I know it's a Monday, it's a busy, busy day in the spring, and I want to thank you for taking the time to join us on this, as always, on our CAP Today webinars. So with that, thanks once again to Drs. Mata, Vemula, Carolyn Rowe, and Fluidigm, and all of you. And with that, I'll bring this webinar to a close. Thank you.